Good day, future doctors. So this is the second part of our series of lectures on immunodeficiency disorders. We will start this lecture with the last two primary immunodeficiency disorders affecting the phagocytes. We have the leukocyte adhesion defect types 1, 2, and 3, and the interferon-gamma receptor deficiency. Leukocyte adhesion defects types 1, 2, and 3 have something to do with the entire process of chemotaxis. Chemotaxis has four phases. You have the margination, rolling, adhesion, and diapedesis. The last three steps, the rolling, adhesion, and diapedesis, would involve the display of proteins by both the white blood cells and the endothelial cells of the wall of the blood vessels. These proteins will help the white blood cells to anchor themselves to the wall of the blood vessel so that it would be easy for them to go out of the blood vessel and go towards the site of infection. In the process of rolling, the phagocyte or the white blood cell will display sialyl-Lewis X protein on its surface, and that's the light blue colored structure that you can find displayed on the surface of the phagocyte in the photo. This phagocyte will use its sialyl-Lewis X to bind itself to the P and E selectins expressed by the endothelial cells. And since the binding of the sialyl-Lewis X to the E and P selectins is weak, then eventually the white blood cell will detach and the white blood cell will undergo the process of rolling. Let me correct something from our previous lectures. I told you in the second part of our introduction to immunology that the proteins involved in rolling are the L selectins of the white blood cell and the E and P selectins of the endothelial cells. Today, the L selectins are only recognized as important in the homing process of lymphocytes. In the case of neutrophils and other phagocytes, it's the sialyl-Lewis X that is used during the process of rolling. Rolling is followed by the process of adhesion. In adhesion, the white blood cell or the phagocyte will express integrin. And this integrin will be used by the phagocyte to attach itself to the ICAM, intercellular adhesion molecule, that is expressed by the endothelial cells. And since the binding between the integrin and the ICAM is very strong, then the cell will no longer roll. Thus, the process is called adhesion. Let's talk about leukocyte adhesion defect type 1. The problem in this disorder is the mutation in the gene that encodes for the protein integrin or CD18. So expect that there would be a problem in the adhesion step of chemotaxis. White blood cells cannot go out of the blood vessel and migrate towards the site of infection. So expect that the sites of infection of these patients will become severe or will progress to extensive level because of the absence of the white blood cells that should have cleared the offending agent. And since the white blood cells cannot go out of the blood vessel, and the bone marrow will constantly produce white blood cells, thinking that the white blood cells will be able to clear off the microorganism, then what will happen is there would be a marked leukocytosis. And that's the reason why some of these patients are mistaken to have leukemia because they have a high WBC count. After the baby is delivered, it's very important to separate the baby immediately from the placenta. That is to prevent blood loss. So what we do in the hospital is we apply umbilical clamp on the umbilical cord, and then we also clamp a portion of the umbilical cord using a forceps. And then we will cut the umbilical cord between the umbilical clamp and the forceps. The remaining portion of the umbilical cord, or you can just simply refer to it as umbilical stump, should fall off from the body of the newborn 2 weeks after the delivery. Why? It is subjected to drying, it is subjected to necrosis, and bacterial colonization. You know very well that if there is the presence of bacteria, there would be recruitment of neutrophils. And every time the neutrophils will try to clear off the bacteria, they can't help but release elastase enzymes. And these elastase enzymes now would contribute to the detachment of the umbilical stump from the newborn. Now, in patients with leukocyte defect type 1, the umbilical stump will not fall off 2 weeks after delivery because of the fact that the neutrophils were not able to go out of the blood vessel and go to the site of the umbilical cord. Worst, since the bacteria are not eliminated by the neutrophils, the area could also have an infection, and we will refer to it as omphalitis. One of the first signs that a newborn could be having leukocyte adhesion defect is the delay in the detachment of the umbilical cord stump. Again, it should fall off within 2 weeks. Failure to do so should raise the suspicion that the child could be having leukocyte adhesion defect type 1. Aside from the delay in the detachment of the umbilical stump or umbilical cord stump, some patients will even develop omphalitis, and that's the infection of the umbilical area. And since this is a case of leukocyte adhesion defect, expect that the sites where there are infection would not present with purulent discharges because of the absence of white blood cells. Next is leukocyte adhesion defect type 2. Compared to type 1, this is rare. The problem in this medical condition is the failure of the white blood cells to express the sialyl Lewis X, which is very important in the rolling step of chemotaxis. And the reason behind why white blood cells cannot express the sialyl Lewis X is that there is a mutation in the GDP fucose transporter, which is responsible for the transport of fucose into the Golgi apparatus. Take note, sialyl Lewis X is a protein, but part of it is fucose. So while the protein is being processed in the Golgi apparatus, fucose should be incorporated to it. And for the fucose to enter the Golgi apparatus, there must be a GDP fucose transporter. This one is absent in individuals with leukocyte adhesion defect type 2. That's why they cannot express the sialyl Lewis X on the surfaces of their white blood cells. You have here a cell. In the center of the cell, in its cytoplasm, you have there the GDP fucose. And that GDP fucose should be transported to the pink-colored Golgi apparatus by the GDP fucose transporter. And that one is missing in the case of leukocyte adhesion defect type 2. So therefore, the sialyl Lewis X protein will never be incorporated with fucose. Thus, it will not be expressed on the surface of the migrating white blood cells, leading to the failure of the rolling step. Take a look at the structure of sialyl Lewis X. The red triangle in there is fucose. Without the fucose, this entire protein will never be displayed on the surface of the affected white blood cell. This is just to make you appreciate that during the rolling step, you have there the expression of the sialyl Lewis X, and that sialyl Lewis X will be used by the phagocyte or neutrophil or white blood cell to attach themselves to the E and P selectins expressed by the endothelium. One more thing that I want you to take note. Leukocyte adhesion defect type 2 is not characterized with delay in the detachment of the umbilical stump. I tried my best to look for a reason why the delay in the detachment of the umbilical stump is only prominent in leukocyte adhesion type 1, but I cannot find a reason. What is associated with leukocyte adhesion defect type 2 is the Bombay phenotype of the ABO blood group. Take note that we have A and B antigens expressed on the surfaces of our red blood cells. If you have A antigen, then you will be called type A. If you have B antigens, then you will be called type B. If you have both of them, then you will be called type AB. But if you don't have any of them, then you will be classified as type O. But before we can express these A and B antigens on the surfaces of our red cells, they must have a precursor. And that precursor is the H antigen. Take a look at the red blood cell in the picture. Attached to it is a group of sugar molecules that are linked together. These sugar molecules will now form the H antigen. Now, to form the A antigen, we'll just simply add to the H antigen N-acetylgalactosamine. So what I want you to understand at this point, without the H antigen, the A antigen will never be formed. Now, how do we form the B antigen? So you should have the H antigen, but instead of adding N-acetylgalactosamine, you will incorporate galactose. The point is, without the H antigen, the B antigen will never be expressed. You are blood type AB if you have both the galactose and the N-acetylgalactosamine attached to the AH antigen. But if you do not have the galactose and the N-acetylgalactosamine attached to the H antigen, then that's the time you will have the blood type O. Type O individuals only have the H antigens on the surface of their red blood cells because they did not inherit from their parents the A gene and the B gene that will allow them to incorporate N-acetylgalactosamine for A antigen and galactose for B antigen. So if you are type O individual, so you can just assume that you have the H antigen. But if you are blood type A, you can assume that you have the A antigen and the H antigen at the same time. Now, take a look at the structure of the H antigen. One of its sugar molecules is fucose. So therefore, during the formation of this H antigen, you also need the GDP fucose transporter that is absent in leukocyte adhesion defect type 2. So what will now happen is the red blood cell will not be able to express the H antigen. So even if the person inherited a gene from his parents, he cannot express the A antigens on the surfaces of his or her red blood cells because they don't have the precursor H antigen. So instead of being classified as type A blood type, the person now will be classified as type O. The real blood type O individuals have H antigens on the surfaces of their red blood cells. And these individuals have anti-A and anti-B antibodies in their plasma. While a patient who has the Bombay phenotype will not have any antigen displayed on the surface of their red blood cells. And what is bad about being a Bombay phenotype is that since you do not have the H antigen, you will have a corresponding antibody against it. That's the characteristic of the ABO blood group. What you do not have, you will have an antibody against it. So for a blood type A individual, the person will have anti-B. For a blood type B individual, the person will have anti-A. In the case of Bombay phenotype, since this person doesn't have the H antigen, that person will also have anti-H. So that's why you will be surprised to encounter a patient who is blood type O, and that patient will reject blood type O blood because there's a probability that the patient or the recipient is a Bombay phenotype, and his or her anti-H are attacking the H antigens of the true blood type O donor's blood. Blood type A individuals have both A and H antigens on the surfaces of the red blood cells, and they have anti-B in their plasma. Blood type B individuals have both B and H antigens on the surfaces of the red blood cells, and they have anti-A antibodies. Blood type AB individuals have the three antigens, the A, the B, and the H antigen, and they do not have any antibodies present in their plasma, or else they will be destroying their own red blood cells. Blood type O individuals, the real ones, the real blood type O, they only have H antigens on the surfaces of their red blood cells, and they have anti-A and anti-B antibodies. In Bombay individuals, they do not have even the H antigen on the surface of their red blood cells, and they can produce anti-A, anti-B, and the most important one, anti-H, which will make them reject all the blood types, including the blood type O. Leukocyte adhesion defect type 3 is very similar to leukocyte adhesion defect type 1. So there would also be delay in the detachment of the umbilical cord stump. There would also be probability of omphalitis. But the problem with leukocyte adhesion defect type 3 is not the absence of the integrin. These individuals can display integrin proteins on the surfaces of their white blood cells. So what is now the problem with leukocyte adhesion defect type 3? It has something to do with the kindlin 3 protein. This kindlin 3 protein is the protein responsible for activating the integrin protein. So without this protein, the integrin will become non-functional. So even if the cells of these individuals can display integrin, their integrin will not help them during the adhesion step. This kindlin 3 protein is also important in the functions of the platelets, especially the time that they are aggregating to form the platelet plugs. The absence of this will put at risk the individual to have bleeding disorders. So that will somehow differentiate leukocyte adhesion defect type 3 from type 1. Shown in the picture is one integrin protein displayed on the surface of the white blood cell. It has two chains, the alpha and the beta. And then I want you to take a look at the cytoplasmic side. You have there one protein that is labeled as kindlin. That's the kindlin 3 protein that is missing in leukocyte adhesion defect type 3. And that protein is involved in the activation of the integrin. Thus, the absence of kindlin 3 will affect now adhesion process of chemotaxis. Kindlin 3 is also important in the function of platelets, particularly during the time that they are forming the platelet plugs. The absence of this protein will increase the chance for bleeding disorder. The picture on the left side of the screen is the normal peripheral blood smear, and shown on the right side of the screen is the peripheral blood smear of a patient with leukocyte adhesion defect. Can you appreciate the significant increase in the number of neutrophils? Why do you think there is that significant increase in the number of neutrophils? Because they are constantly produced by the bone marrow because of the unending chemotactic stimuli from the site of infection. The problem is, even if the bone marrow can produce a lot of neutrophils, these neutrophils cannot go out of the blood vessel. That's why you see them in significant numbers in the peripheral blood smear. Tissues that are infected, especially with bacteria, will be infiltrated by neutrophils. So the nucleated cells that you can see in this particular tissue are actually neutrophils. Please refer to the left photo. The three arrows are pointing to areas within the tissue that are infected with bacteria. But what do you notice? Those areas were not infiltrated by neutrophils, signifying the failure of the neutrophils to complete the process of chemotaxis. Then refer to the right photo. You have there in the center a blood vessel, and the clue is the presence of red blood cells. Pointed by the two arrows are the neutrophils that are trying their best to go out of that blood vessel to go to the site of infection, but they cannot do so because they lack either integrin or the sialyl Lewis X. In some cases, especially in leukocyte adhesion defect type 3, they have the integrin, but the integrin is not working because of the absence of the kindlin 3 protein. One of the laboratory procedures that we can perform to diagnose cases of leukocyte adhesion defects is flow cytometry. In this particular instrument or machine, the cells are made to flow in a single file because of the principle of hydrodynamic focusing. That's why you can see in the picture, the cells are flowing in a single file. And at a certain portion along the instrument or machine, the cells are intercepted by a laser light. And you know what will happen? Each of the cell will emit a certain type of fluorescent light. I know some of you are wondering, how can a human blood cell emit fluorescent light? The answer is, before the cells are made to flow inside the flow cytometer, they are tagged with antibodies carrying fluorescent dyes. So what will happen is, if the cell will be hit by the laser light, the fluorescent dye will produce color. And the color now will make us identify the type of cell. How? Before the cells are made to enter the fluorometer machine, the cells are first mixed with antibodies that are labeled with fluorescent dyes. If you want to identify the cells, you will use specific antibodies. And these antibodies should carry specific fluorescent dye. So, let's say for example, for neutrophils, you will use an antibody that carries a blue fluorescent dye. For, let's say, basophils, you will use antibodies that would carry yellow colored dyes. Now, for eosinophils, you will use antibodies that would carry red fluorescent dyes. So, what will happen is, if the light will hit a neutrophil, the fluorescent dye will emit blue color. And that will now be interpreted by the flow cytometer as the signal coming from a neutrophil. But if the cell will emit red fluorescence, then that will give the flow cytometer the idea that the cell that is currently hit by the laser is an eosinophil. So how can we use now flow cytometry to diagnose cases of leukocyte adhesion defect? I will use LAD type 1 as an example, and that's the absence of integrin, right? So what will I prepare in the laboratory are antibodies to integrin. Then I will label those antibodies with, let's say, blue fluorescent dye. So I will add those anti-integrin antibodies into the sample of the patient. So if the patient has integrin proteins on the surfaces of their white blood cells, these antibodies will bind to them. So that if the cell will pass through the flow cytometer and if it is hit by the laser, the cell will emit blue fluorescent light, indicating that the patient has integrin proteins and the patient doesn't have leukocyte adhesion defect type 1. But If the white blood cell will not emit blue fluorescent dye after it is hit by the laser, that would only mean that the white blood cell does not have, antibodies to integrin attached to it, because in the first place, it doesn't have integrin. And that will now give us an idea that the patient sample is positive for leukocyte adhesion defect type 1 or the absence of integrin or CD18. Just like in the case of Chiari-Higashi and chronic granulomatous disease, there is no treatment for leukocyte adhesion defect. So as future doctors, we can manage the patient with antibiotics once we will identify infection. We can also transfuse granulocyte concentrate, which I will explain later on. And for the patient to fully recover and will not have any recurrent infections anymore, we can go for bone marrow or hematopoietic stem cell transplant. But the problem is this is very expensive. Another problem is that the patient or the family of the patient has to look for possible donors. But these procedures offer very high success rate. In blood banks, we usually collect 450 to 500 ml of blood from donors, and we refer to that as whole blood. It's whole blood because you have there the plasma, the red blood cells, the platelets, and the white blood cells. Now, I want you to imagine if we will give 6 blood bags to a recipient, we're actually giving 3 liters of blood because one blood bag is 450 to 500 ml, and that can cause circulatory overload. That's why it is best to only give the patient what the patient needs. In the case of leukocyte adhesion defect patients, they only need functional white blood cells. So there's no need for us to give the platelets, the plasma, and the red blood cells. That's why in blood banks, we usually process blood and separate its components. That's why there is what we call granulocyte concentrate, which can be given to patients with leukocyte adhesion defects. One of the procedures done in blood banks to prepare granulocyte concentrate is apheresis. Blood is drawn from the patient. The blood is made to flow through the apheresis machine, and the apheresis machine will separate the white blood cells, particularly the granulocytes, from the blood of the patient. The remaining parts of the blood will be then again returned to the patient. So the machine now, or the apheresis machine, has already collected granulocytes from the blood of the patient, and that will now be called as granulocyte concentrate. Again, in the apheresis procedure, blood is drawn from the patient. Blood is separated into components by the centrifuge machines inside the apheresis. The needed components are separated, and the unused components are returned to the donor. The last primary immunodeficiency disorder affecting phagocytes is interferon-gamma receptor deficiency. For you to understand the importance of interferon-gamma, I want you to take a look at this picture. You have here the red-colored antigens engulfed and processed by the yellow-colored phagocytes, and then the phagocytes will present the antigen to the blue-colored T-delayed hypersensitivity or T helper 1 cells. And then in the picture in the middle, you have there the production of the cytokines, particularly interferon-gamma. And interferon-gamma is represented by the 3 triangles in the picture. And that interferon-gamma will go to the blood and will recruit more macrophages and T cells into the site where the antigen was encountered. You have here in this picture an antigen-presenting cell. Inside its cytoplasm, we have there the red circles representing the antigen. And then this antigen-presenting cell is using the MHC class 2, yellow-green in color, to present the antigen to T-delayed hypersensitivity or T helper 1. Also shown in the picture that this antigen-presenting cell will enhance the activation of the T helper 1 by producing interleukin 12. In return, once the T helper 1 will be activated, the activated T helper 1 is blue-green in color. That activated T helper 1 will produce interferon-gamma. And do you know the effect of the interferon-gamma? It will enhance the phagocytic ability and the ability of the APC to present antigens so that it can process more antigens. And for that interferon gamma to exert its effect, the antigen presenting cell must have a receptor to it. But there are people born with no receptor to interferon gamma. That's why their blood cells or phagocytes or antigen presenting cells will not respond to interferon gamma. Interferon-gamma receptor deficiency is also known as the Mendelian susceptibility to mycobacterial diseases or MSMD. Why? Because these individuals are normally resistant to most microbes except for the ones that belong to the Mycobacterium species such as what? Mycobacterium avium, Mycobacterium fortuatum, and Mycobacterium kansasai. These microorganisms can cause skin infection, bone infection, joint infection, and these Mycobacterium species do not normally cause disease among immunocompetent individuals. Another problem with MSMD is that administering Bacille Calmette Guerin, a vaccine that is supposed to prevent Mycobacterium tuberculosis, can cause the patient to develop an infection. This BCG contains live attenuated Mycobacterium bovis, meaning weakened Mycobacterium bovis. Even if the Mycobacterium bovis was already weakened, that bacterium can cause disease in a person with interferon gamma receptor deficiency or Mendelian susceptibility to mycobacterial diseases. We will now move to the primary immunodeficiency disorders involving the B cells. You have the Brutons X-linked Agammaglobulinemia. You have the CVD or Common Variable Immunodeficiency. You have the immunodeficiency with hyper-IgM. And you have the Selective IgA deficiency. Take a look at this pie chart and appreciate that majority of the cases of primary immunodeficiency disorders that are encountered in clinics and hospitals are the ones involving the B cells, 50%. Let's start with Brutons X-linked Agammaglobulinemia. From the name of the disorder, it's very obvious that it will manifest among males because males only have one X chromosomes and their mothers are referred to as carriers because even if the mother has the abnormal gene, the other X chromosome can actually compensate. Bruton's X-linked Agammaglobulinemia is characterized by the failure of the B cell precursors to become the mature B cells. And the main reason behind it is the absence of the Bruton's tyrosine kinase enzyme because of the mutation of the gene that encodes for that enzyme in the long arm of chromosome X. And that's the reason why this disorder is named X-linked Agammaglobulinemia. Agammaglobulinemia means there is no production of antibodies because you don't have mature B cells to become the plasma cells. Now, how important is the missing tyrosine kinase enzyme? It is important in the maturation of the B cell precursors. Shown in the picture are the immature B cell precursors. And for them to progress from one stage to the other, they need the Bruton's tyrosine kinase enzyme, which is absent in X-linked Agammaglobulinemia. The clinical signs and symptoms of Bruton's X-linked Agammaglobulinemia will start to appear as soon as the child will reach the age of 7 months. Why? Because for the first 6 months of life, we are protected by the maternal IgGs which crossed the placenta. And since that was just a form of natural passive immunity, the IgGs that we got from our mothers will be eventually consumed and they will reduce in number because of their half-lives. So expect that as we reach the age of 7 months, we should be able to produce our own antibodies. But the problem is, patients with Bruton's X-linked Agammaglobulinemia do not have mature B cells to become plasma cells and produce antibodies. Again, children with X-linked Agammaglobulinemia are usually healthy for the first 6 months of life because of the transfer of maternal IgGs through the placenta. In children with X-linked Agammaglobulinemia, infections generally take longer to get better or to recover, and then they come back again even if the patient was managed with antibiotics. I hope you can remember from the part 2 of our lecture on Introduction to Immunology that we can actually run this test to help us with the diagnosis of Bruton's X-linked Agammaglobulinemia. What are the expected results again? For electron microscope, you will not be able to demonstrate the presence of hairy cells. Why? Because mature B cells appear hairy under electron microscope because they have surface immunoglobulins. And since the B cell precursors cannot reach the mature B cell stage, then expect that the electron microscope procedure will yield a negative result. Next, CD19, CD20, and CD21 are CD markers of mature B cells. If you cannot demonstrate the presence of these markers in the lymphocytes of the patient, then that would only mean that the patient doesn't have mature B cells. And that is highly suggestive of Bruton's X-linked Agammaglobulinemia. And from the name of the disease, Agammaglobulinemia, expect that there would be low antibody levels or titers in the serum or plasma of the patient. We can also utilize flow cytometry to determine whether the patient's blood has mature B cells. So we will mix the blood of the patient with antibodies to CD19. That's one of the CD markers of B cells, right? We will make it a point that the antibody to CD19 is labeled with a fluorescent dye. So let's say, for example, the fluorescent dye is supposed to produce blue color. So that if the B cell of the patient will be intersected by the laser light source, that B cell will be able to produce blue fluorescence. The absence of blue fluorescence would mean that the antibodies were not able to bind to CD19 because they are not present in the cells of the patient because the patient doesn't have mature B cells. Compare now the two results of flow cytometry. One shows you the detection of the fluorescent light produced by the CD19 antibodies labeled with the fluorescent dye. And then take a look at the result of the X-linked Agammaglobulinemia patient. There is no detection of fluorescent light formation by the cells of the patient, indicating the absence of CD19, thus absence of mature B cells. Since the problem with Bruton's X-linked Agammaglobulinemia is the low antibody titer, treatment would include administration of immunoglobulin, or you call it as immune globulin replacement therapy. And it's also very important to give the patient antibiotics. The next primary immunodeficiency disorder that is affecting the B cells is the Common Variable Immunodeficiency or CVID. Just like in the case of Bruton's X-linked Agammaglobulinemia, patients with Common Variable Immunodeficiency will also have low antibody titers or levels in their plasma. Then, expect that these patients will have recurrent bacterial infections, particularly encapsulated bacteria. The exact cause for CVID is still unknown, but most scientists are suggesting there is a failure of the mature B cells. So meaning these patients have mature B cells. There's a failure of the mature B cells to differentiate into plasma cells. So in Bruton's X-linked Agammaglobulinemia, there is a failure to become the mature B cells. In CVID, there are mature B cells. It's just that they do not differentiate to form the plasma cells. So what are now the expected results of these tests in a patient with CVID or Common Variable Immunodeficiency? Remember, they have mature B cells in their blood. It's just that they do not form the plasma cells. So expect that electron microscope will demonstrate hairy cells because these patients have mature B cells. They will also be positive for CD19, CD20, and CD21 because again, they have mature B cells. Just like the case of X-linked Agammaglobulinemia, they will have low antibody levels in their plasma. So we're done with Bruton's X-linked Agammaglobulinemia and Common Variable Immunodeficiency. It's now time to discuss immunodeficiency with hyper-IgM. From the name of the disorder, expect that the patient would have high levels of IgM but low levels of the other isotypes of antibodies. So expect that there's a problem with the process we call as class switching. You have here in this picture, left side, the antigen presenting cell presenting the antigen to T helper cell. And on the middle of the picture, you have here an activated T helper cell that is activating a B cell to become plasma cell. Now, the first antibody that will be produced by the plasma cells is IgM. But you know very well, this IgM should be later replaced by IgG and the other isotypes of antibodies. So, you call that process as class switching. How is class switching made possible? Take a look at the activated T helper cell and the B cell. Please take note of the CD40 ligand displayed on the surface of the T cell. And that CD40 ligand has a corresponding CD40 receptor present on the B cell. Now, for the B cell to class switch from IgM to IgG and other types of antibody, you need the interaction of CD40 ligand and CD40 receptor. CD40 ligand is also known as CD154. Immunodeficiency with hyper-IgM is characterized by the mutation in the gene that encodes for the CD40 ligand or CD154. Please recall that this CD40 ligand is expressed by the T cell so that it can activate the CD40 receptor present in the B cell, and it will make the B cell do the class switching from IgM to the other isotypes of antibodies, particularly IgG. Now, the mutated gene is found in the X chromosome. So therefore, this disorder is an X-linked type. In the absence of the CD40 ligand, there is no class switching. So expect that the patient would have low levels of IgG, IgA, IgD, IgE, but they have a normal or high levels of IgM. Again, the problem with hyper-IgM is the absence of the CD40 ligand, which is expressed by the T helper cell and used by the T helper cell to make the B cell switch from IgM to IgG and other isotypes of antibodies. In immunodeficiency with hyper-IgM, there is that failure to instruct the B cells to switch their production from IgM to IgG to IgA to IgD and to IgE. That's why expect that the lab results will demonstrate high levels of IgM but with low levels of IgG, IgA, IgD, and IgE. These individuals would have normal B and T cell counts because the only problem here is the failure of the expression of the CD40 ligand. One test that we can also perform is flow cytometry. We will make use of antibodies against the CD40 ligand with labeled fluorescent dye. So these antibodies will bind to the surface of T cells because they are the ones expressing the CD40 ligand. So if the T cell is intercepted by the laser light, that T cell should produce a fluorescent light. If not, then that would only mean that that T cell doesn't have the CD40 ligand. We can utilize flow cytometry in the diagnosis of hyper-IgM. We will just simply use antibodies to CD40 ligand. These antibodies are coupled with the fluorescent dye. So expect that this anti-CD40 ligand will bind to the CD40 ligand expressed by the patient's T cells. So if these T cells will be intersected by the laser light, they should produce fluorescence. If not, that would mean that the CD40 ligand is absent and the patient is a case of immunodeficiency with hyper-IgM. Majority of the patients with hyper-IgM would have recurrent infections on the upper and lower respiratory tracts caused by the encapsulated bacteria. Why? Because these patients have low levels of IgG, and IgG is an opsonin. Do you know patients with hyper-IgM would also present with severe viral infections? Why again? Because viruses are fight off by the immune system via neutralization. And neutralization is best facilitated by IgG. And IgG levels are very low in patients with hyper-IgM. We will now discuss the last primary immunodeficiency disorder affecting the B cells, and this is the selective IgA deficiency. Please take note that IgA has two forms. It has the monomeric serum IgA and the dimeric secretory IgA. Patients with selective IgA deficiency has low levels of both IgA1, serum IgA, and IgA2, secretory IgA, but they have normal levels of the other antibodies. And the mechanism behind this one is still unknown until today. The only explanation that scientists can provide is that these patients have mature B cells. The problem is these mature B cells will never differentiate into IgA producing plasma cells. Most patients are asymptomatic. Should they become asymptomatic, there would be recurrent infections in the respiratory, gastrointestinal, and urogenital tracts. Why? Because these areas in the body are rich in secretions, and that is where you can find the secretory IgA. Without the secretory IgA, expect that there would be recurrent infections on those areas. This is very important to discuss because you will encounter this one in immunohematology or blood banking. Do you know that patients with selective IgA deficiency can develop a type of transfusion reaction, and we call it as Anaphylactoid transfusion reaction? So from the name of the condition, no, you can already predict that it has something to do with histamine. Now, what happens here is if a patient with selective IgA deficiency will receive blood products from normal individuals, they will encounter now IgA for the first time because they don't have it in their blood. So the immune system of these patients will consider the IgA as foreign. So the immune system of the patient will produce antibodies. Most of the time, the antibodies produced are IgE type because IgA is a potent allergen according to some of the references. That's why patients with selective IgA deficiency will be producing IgEs against the IgA. If the patient will receive another blood product in the future, the IgA of the donor will now cross-link the IgEs present on the mast cells and basophils, and the patient will suddenly complain of pruritus, difficulty of breathing, and other allergic associated signs and symptoms during the blood transfusion. Thank you very much for listening. That ends our part 2 of immunodeficiency disorders. In the next video, we will be discussing the primary immunodeficiency disorders affecting the T cells and the primary immunodeficiency disorders affecting both B cells and T cells. So you should look forward to learning them as well.